Hello everyone. So I am here to discuss the extraction and isolation of various chemical compounds, especially phytochemicals that can be obtained from plant species. Okay, it could be known compound means already discovered and reported compound. It or else it could be unknown compound that could have some bioactivity in vitro and in vivo as well, that could be further utilized to discover the drug. Okay, so as to use as medicine. So basically our focus is to drug discovery from natural products from plant species, animal species, or some other organisms that could be useful for new drug discovery or new chemical moiety for those diseases which do not have specific or proper drug till now. So here, so first of all, what we need to do, the plant part we need to take, in which the targeted drug molecule or the chemical molecule is present in a rich amount. Okay, that plant part is further dried fully for moisture removal. Okay, so first drying is done, then it is grinded. Grinding is done means that plant part is further subjected to break into smaller parts, smaller fragments. Okay, so so the purpose is to increase the area of contact, because we are going to subject this material, this grinded material, to a specific solvent, that the molecule present inside that plant part will get dissolved in the specific solvent which we will use. Okay, so that's why to increase the area we do grinding. Okay, after that, this is the basic extraction part. So these are grinded, already grinding is done, drying and grinding is done means small parts are there, so at larger parts made to smaller particle like structures. Okay, mm, maybe almost in the powder form. Okay, so it could be 1 kg or some amount, or 500 for 500 gram. Yeah, okay, so now these these are dissolved in a specific solvent. Okay, so if we dissolve these in a specific solvent, then the process is usually maceration. But if we use the safe if we use that heat. Okay, so much amount means higher temperature means it will facilitate the release of drug molecule or chemical molecule present inside the coarse particle or plant material to release into the solvent. Okay, so that could be another process. Okay, so extraction is done. So this extracted amount is having reached means, other than the targeted chemical compound targeted chemical compounds are there, other than also some other compounds are also there in the extraction. Okay, so now this extraction is further subjected to some form of partitioning. Okay, so partition, what it does in partitioning, like you may say, like in extraction we have used methanol, so it is polar, or maybe ethyl acid, it is also polar, but more polar is methanol. So in case we use methanol for the extraction, first step in maceration or some other form of extraction, so this means more polar compounds will get dissolved in the methanol extract. Okay, so methanol extract will have very high amount of polar compounds. So now these polar compounds, many type of polar compounds may be there. So we need maybe flavonoid, for example we need flavonoids. So in case of flavonoid, so we need to make flavonoid rich fraction. So for this we can further subject this methanol extract to partitioning by doing like 9:1 water is to methanol ratio. Okay, so in 9:1 water methanol ratio we can put this methanol extract to dissolve, okay, properly. And now this this dissolved part is further subjected to ethyl acetate and n-butanol partitioning. Means ethyl acetate is less polar, n-butanol is very polar. So very polar means flavonoids rich fraction. Flavonoid means it could have some form of sugar quantity. Okay, so glycan part may be there. So if glycan part is there, means glycosidic linkage is there. So glycosidic linkage will certainly lead to more polarity to in the compounds. So these compounds will be more dissolved in the n-butanol fraction. Okay, so n-butanol fraction. So now n-butanol fraction we got this n-butanol fraction may be further subjected to RP flash chromatography, reverse phase flash chromatography. So it leads to fractionation of those flavonoid rich fractions. Means now maybe that that n-butanol fraction may have many type of compounds. Means 10 or 15 type of compounds may be present. Okay, much polar, but when we fractionate it by the process of RP flash chromatography, then it could have each fraction 2-3 compounds or 3 to 4 compounds. Okay, so it is highly enriched and separated well. So this separation can be used further. Okay, so these 3-4 type of compounds may be further subjected to normal type of chromatography. Okay, or some form of cephatics. So in case of cephatics, it is size exclusion chromatography. What it does, the much molar the molecule having higher molecular weight will elute fast in the cephatics LH20 column. Okay, so means glycosidic linkage is there. If some compound has glycosidic linkage, so that compound may have higher molecular weights, so that it can elute fast in the cephatics LH20. Okay, lesser molecular weight containing molecule will elute later. So now this by this cephatics we can easily purify the targeted compound. Okay, so now this purified compound may be further subjected to HPLC analysis for proper analysis, for very appropriate analysis. Means maybe in TLC it is showing only one spot, means it is pure, maybe it is showing, but when we take this sample or this compound to HPLC analysis, it could show 2 spots or 2 peak. Means maybe other compound is also present. Not necessarily always it will happen, but in some cases it happens, because HPLC is very high performance chromatography, right? It is the name, high performance liquid chromatography. So performance is high there, separation is much more. Okay, so if HPLC done, then if this is a purified compound, then we can characterize these compounds by mass spectrometry. By mass spectrometry we can know the molecular weight of that compound. LCMS by purifying, and mass spectroscopy, and NMR. For NMR, in case of NMR, this shows the characterization, most useful characterization parts. Okay, NMR, by NMR spectroscopy we can compare this NMR data to the standard NMR data available in the literature. Or if it is unknown compound, then we need to do many type of NMR, means 1D NMR, okay, 2D NMR likewise. Okay, so we need to do many type. And in case we need to know the functional group, then we can do IR, infrared FTIR, Fourier transform IR infrared spectroscopy, or simply IR spectroscopy. Okay, so by this characterization can be done. So it can lead to some known compound or some unknown compound that could have a bioactivity that could be used later as drug. Okay, thank you.